This experiment is designed to identify lipid-binding proteins and to analyze lipid-protein interaction in a defined membrane environment of lipid vesicles. Proteins of cell homogenates are incubated with the freshly prepared lipid vesicles from phosphatidylserine and ceramide. The reaction mixture is then supplemented with Annexin V-conjugated magnetic beads to anchor the protein associated with the lipid vesicles to the magnetic beads. Next, protein-lipid vesicles are purified by magnetic activated cell sorting to identify the lipid-bound protein. Demonstrated results use SDS-Page Western blotting to show LIMACS purification of endogenous and recombinant atypical PKC and a specific ceramide binding domain expressed in MDCK cells.
The main advantage of this novel technique over existing methods like ELISA or plasma resonance spectroscopy like surface binding is that the lipid-protein interaction is achieved in a membrane-like environment and that the binding proteins can be purified and analyzed. The implications of this technique extend toward therapy and diagnosis of diseases involving lipids, because in these diseases the physiological lipid-protein interaction is impaired. For some of these diseases, the protein targets are not even known and need to be identified to design a lipid-based therapy.
Begin with lipid vesicles prepared from a dried mixture of equimolar amounts of phosphatidylserine and C16 ceramide in 100 microliters of vesicle buffer. Sonicate for 1 hour. After adding 300 microliters